Hello Bio 100. This particular lecture will cover the second half of Chapter 9, and if you recall, Chapter 9 covers meiosis. Meiosis is the second type of cell division we talked about, mitosis being the first. Meiosis, of course, is the process of cell division that's involved in creating the gametes, the eggs and the sperm. Meiosis cuts the chromosome number in half, and so it takes a cell from being diploid to being haploid. In other words, you have one copy of each homologous pair of chromosomes at the end of meiosis. Additionally, what we see is that meiosis introduces genetic diversity into offspring. Genetic diversity is extremely important in terms of the preservation of a population. The more diverse a population is, the greater the ability that population has to adapt to a changing environment. So what we notice is that in the case of meiosis, there are two processes that introduce this genetic variation. Number one, crossing over, and number two, independent assortment. We begin with crossing over. So crossing over is a process that happens in prophase 1 of meiosis. In other words, the prophase of meiosis 1. And in this particular process, we see, as depicted on this particular slide, that the homologous pairs of chromosomes pair up with one another in this process referred to as synapsis. And so if I use a little pointer on the screen, we see that we have a homologous pair of chromosomes, and each chromosome, of course, has been duplicated. So what we see is that as I point to, with the pointer there, I'm pointing to one sister chromatid of the blue chromosome, and this is the other sister chromatid of the blue chromosome. If you recall, sister chromatids are identical. They're exactly the same. There's no difference in the genetic material that's present in sister chromatids. And so, of course, we also have a sister chromatid for the maternal chromosome. And I'm randomly assigning these maternal and paternal based on color. It could be the other way, but your book is drawing them in different colors to illustrate the fact that one of these came from this organism's mother. Let's say the red one came from the mother, so it's the maternal chromosome, and one came from the father. You have to think back to the first half of this particular chapter and the fact that all organisms are, well, not all organisms, but at least humans and many organisms are diploid. And being diploid means that you have two copies of every single chromosome, and those chromosomes contain alleles. Recall that alleles are alternate forms of a gene. This is important for understanding what's going to happen now in crossing over. So if we follow this diagram from our text to the right, what we see is this, that these sister chromatids reach out and they swap segments. And so what we're left with, if we're following the pointer that I'm using, we're now in the third little stage of this diagram. We now have a blue chromosome, which we were calling the paternal chromosome. So all the blue chromosome came from the father, but the red chromosome came from that person's mother. And so the chromosome is now mixed. It contains DNA from two different people. If we look over at the red, of course, it's mostly maternal, but a little bit paternal. And so in this manner, the genes are scrambled. You realize, if you understand meiosis, and it's very important, of course, that you do, that when the egg or sperm is created, it's going to get either the paternal chromosome, in other words, one of these blue ones, or it's going to get the red one. And so that means that the chromosomes that are passed down into the gametes are not identical, but rather they consist of DNA that has been scrambled as a result of this process of crossing over. If we follow our diagram to the right, then what we notice is that on the far right here, we have undergone meiosis 2. And so after meiosis 2, you recall that the sister chromatids split. So at the end of meiosis 2, we have 1, 2, 3, 4 chromosomes all lined up there. Those 4 chromosomes are going to go to 4 different gametes. And in this particular case, this first one is all paternal. The second one is mostly paternal, a little bit maternal. The third one is mostly maternal, a little bit paternal. And then the last one is completely maternal. So crossing over is an event that occurs each time meiosis takes place. And furthermore, crossing over is random. The point at which a break occurs in a chromosome is random. And so it's this random scrambling of the genes that mixes the genes up before they're passed on to the next generation. This, therefore, increases the amount of genetic variation that's found in the offspring. The second mechanism that introduces genetic variation into offspring is referred to as independent assortment. And so independent assortment is the process by which the chromosomes line up at the metaphase plate in metaphase 1. And so remember, a common exam question in this material is, what lines up at the metaphase plate in metaphase 1, metaphase 2, and mitosis? And of course the answers are, in mitosis, the chromosomes themselves line up at the metaphase plate and the sister chromatids are going to split. And that same thing happens actually in meiosis too. But in meiosis 1, the homologous pairs of chromosomes line up at the metaphase plate and the homologous pairs are going to split. So if we look at the diagram on the screen from our text, then this really illustrates to us how independent assortment works. Now, if we just have 3 pairs of chromosomes, we have an organism that has 3 pairs of chromosomes, so in other words, they have a total of 6 chromosomes, then there are 8 different ways those chromosomes can line up. If we start at the left-hand side, what we see is that it's possible that all the paternal, in other words, the blue chromosomes, could line up on the left. It's also possible that all the maternal chromosomes, in that same example, the maternal chromosomes would line up on the right. Okay, but then if we go to number 2 here, so the one I'm circling with the pointer. So in this case, we have 2 paternal chromosomes on the left, 2 on the right, and then the last pair is swapped here. And so when they split, they're going to, of course, go to different cells at the end of meiosis 1. And this cell is going to end up with 2 paternal chromosomes and 1 maternal. This cell is going to end up with 2 maternal and 1 paternal. And furthermore, if we make our way across the screen, we see that all the alignments, the 8 that are shown on this screen, are all possibilities for alignments for an organism that has only 6 chromosomes. What this means is the following: that you can calculate the number of possible chromosome alignments by taking 2 and raising it to the number of pairs of chromosomes the organism possesses. In other words, in this particular case, the organism has 3 pairs of chromosomes. So if we take 2 and raise it to the 3, then we have 2 times 2, which is 4, times 2, which is 8. So that's 2 raised to the third, the third power. 2 times 2 times 2 gives us 8. Now, if we look at humans, we of course have 23 pairs of chromosomes. And so if we want to figure out the number of possible alignments for humans, we have to take 2 raised to the 23rd power. And if we do that, what we see is that 2 to the 23rd equals 8,388,608. This is the number of possible alignments that exist when the human chromosomes align as a result of independent assortment in metaphase of meiosis 1. So that's a staggering amount of genetic diversity that's introduced into the offspring at that particular, at least into the gametes and then the offspring at that particular point. So the odds that you would end up with the same alignment in any given cell would be 1 out of 8 million, roughly 400,000 there. And that doesn't even take into account 1 crossing over event. If you add 1 crossing over event, the amount of variability goes up tremendously. It's a massive number, over 1 trillion. And that's just 1 crossing over event. And so crossing over occurs in every single chromosome every time meiosis takes place. And so you can see that there's a tremendous amount of genetic diversity introduced into the gametes and therefore the offspring every time meiosis occurs. And so therefore together, crossing over and independent assortment are very powerful mechanisms that introduce genetic variation into offspring. This, of course, allows natural selection a wide variety of phenotypes on which to work, and this allows evolution to occur. We'll be talking very soon about evolution. So, just to review, what we notice is that meiosis involves two phases, meiosis 1 and meiosis 2. And this, of course, means that in meiosis 1, we have prophase, metaphase, anaphase, telophase. And in meiosis 2, we also have prophase, metaphase, anaphase, and telophase. In other words, each division is broken down into those 4 phases. On this slide, we review the main events of meiosis 1. What we notice is that if we begin on the left-hand side of the slide, we see the chromosomes coming together to form these things called tetrads. This is the point at which crossing over occurs, what we've discussed over the past few slides. And aside from that, prophase 1 looks much like prophase that we observed in mitosis. The spindle apparatus is forming, as you see depicted in the slide, and the nucleus is going away. We now go over to metaphase 1. And so in metaphase 1, what we see is that the chromosomes have aligned themselves on either side of the metaphase plate. So a frequent question, as I mentioned previously, is what lines up at the metaphase plate of metaphase 1? And the answer, of course, in this case is homologous pairs of chromosomes line up at the metaphase plate of metaphase 1. The metaphase plate, if you need reminding, is that imaginary line that goes through the middle of the cell. It's not an actual structure, but it's simply a plane or a line that goes to the center of the cell, and the homologous pairs of chromosomes in metaphase 1 line up on either side of the metaphase plate. In anaphase 1, the chromosomes are pulled apart. Now, of course, the sister chromatids are not in anaphase 1. You'll notice that. The homologous pairs of chromosomes are pulled apart in anaphase 1. Homologous pairs are pulled apart. And then in telophase, telophase 1, what we see is that we end up with 2 brand new cells, and it is at this point we have gone from diploid to haploid. We can tell that from our diagram by seeing this. 2n, of course, means the cell is diploid. This means 2n equals 4. The diploid number of chromosomes, in other words, equals 4. Over here, we have n equals 2. So the haploid number of chromosomes is 2. You can see we have 2 cells. Each cell has 2 chromosomes. Therefore, the haploid number of chromosomes is 2. You'll notice that these chromosomes you can distinguish one from another by their length. So there's a long chromosome and a short chromosome in this cell, and a long chromosome and a short chromosome in this cell. They have separated according to the homologous pairs. Each homologous pair split, and you have one homologue from each homologous pair in each of the brand new cells. We now look at meiosis 2. So in meiosis 2, we're going to see something happen that looks very, very similar to what we saw in mitosis. So in this case, what we notice is that in prophase, the nucleus is disappearing, the spindle apparatus is forming, and look what happens in metaphase 2. In metaphase 2, the chromosomes line up along the metaphase plate. So again, pay close attention to the wording. The chromosomes themselves are lining up along the metaphase plate in metaphase 2. I'm going to jump back a slide, not the homologous pairs. In metaphase 1, the homologous pairs are lining up in the metaphase plate. In metaphase 2, the chromosomes themselves are lining up along the metaphase plate. In anaphase, the chromosomes themselves are splitting. So in other words, the sister chromatids separate. They're pulled apart in anaphase 2. The sister chromatids separate. And then in telophase, we end up with 2 new cells that have been formed from each of the cells that are undergoing meiosis 2. And each of these cells are still haploid. So we began this process of meiosis 2 as haploid, and we finished as haploid. So just to review, as we look back and compare meiosis and mitosis, what we notice is this. In the case of mitosis, we begin with a diploid cell and we end with diploid cells. What happens in mitosis is that in metaphase, all the chromosomes align along the metaphase plate and the sister chromatids are pulled apart, and we end up with 2 cells that are genetically identical, and they are both diploid. They're still diploid. Now, you will notice that the chromosomes themselves are no longer duplicated, right? They no longer consist, if you look up here, if you follow the pointer, this chromosome consists of 2 sister chromatids, and down here, it only consists of 1. That's because the sister chromatids were separated. That's what allows mitosis to occur. and maintain the genetic integrity of the cell. But in meiosis, it's completely different. We have 2 divisions to begin with instead of 1 division in mitosis. And in meiosis, in metaphase 1, the homologous chromosomes, as we see here, and as we stated previously, line up at the metaphase plate. They separate in anaphase 1, and at the end of telophase 1, we have a haploid cell. We've gone from diploid here to haploid there. And then what we notice is that we undergo a second division, which separates the sister chromatids and gives us 1, 2, 3, 4 cells. These cells are not genetically identical to the parent cell for 2 reasons. One is they have half the genetic material that the parent cell did. They're now haploid because the homologous chromosomes have separated. If you're confused, look over at this cell from mitosis. There are 1, 2, 3, 4 chromosomes there. Right? There's 2 of each homologous pair. But if we look over here, there's only 2 chromosomes. So there's 1 of each homolog. 1/2 of each homologous pair has ended up in these 4 cells. But the second reason they're not genetically identical to the parent cell is that we've had crossing over occur, as we talked about earlier in our chapter. And so that means that the chromosomes are all mixed up. So you can see that right here. So this one's red, indicating maternal, 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 and then the end of it is paternal. So the last little bit of it is paternal. So this concludes our 2 chapters on mitosis and meiosis. You want to make sure that you review the differences between them, understand what happens in each stage, and be certain you can answer the question, what lines up at the metaphase plate of mitosis, meiosis 1, and meiosis 2.